The UConn Crumbling Foundation research team presents a basic introduction to the method we have developed for sampling and analyzing concrete samples to determine the presence of pyrrhotite. Additional details can be found in the journal publication posted on our website. We obtain concrete samples by using a 1-inch diameter vacuum drill. The sample is collected as powder in a container attached to the drill and transferred to a plastic bag for transport to the lab. The small hole that is left behind, as you see in the bottom left image here, is then filled with a rapid set concrete patching material, as seen on the bottom right. The whole process takes about 5 minutes and is repeated at least 4 times in different locations of the wall and once in the slab to get representative samples of the whole foundation. There are a couple of reasons why the team has adopted this method. The first one is that obtaining a core is more time-consuming and invasive to the foundation itself. As a result, only 2 locations in the foundation can be sampled at a time. Once the core samples are obtained, further processing is required to obtain a homogeneous powder for the analysis we conduct, including crushing the whole core sample and sieving. Both of these are labor-intensive and time-consuming. In our process, the powder is directly obtained with a drill, and we sample several locations at once to capture a representative section of the foundation. To further discuss our method, let's review the fundamentals of sulfur chemistry, as presented in our Intro to Homeowners video. There are 2 forms of sulfur in concrete, sulfide and sulfate. Sulfide reacts with oxygen when that is present to form sulfate. In concrete, sulfide may be present in the form of pyrrhotite in the aggregate. Cured concrete contains 3 sources of sulfur. Sulfate that comes from the gypsum in Portland cement, sulfide that is still present in the unreacted pyrrhotite in the aggregate, and may also contain additional sulfate from the pyrrhotite that has already oxidized to form sulfate. We utilized 2 methods to determine the different types of sulfur present in the concrete sample. The elemental analysis provides us with a total amount of sulfur in concrete, including both sulfide and sulfate. Then, X-ray fluorescence analysis yields 2 additional, independently obtained results: the ratio of sulfide to total sulfur and the ratio of sulfate to total sulfur. We can then determine the total sulfide present in the sample by combining the 2 methods and multiplying the total sulfur with a sulfide to total sulfur ratio. The concentration we report is grams of sulfide per kilogram of the concrete sample. The amount of measured sulfide can be translated to the total amount of pyrrhotite by considering the proportion of sulfur in the mineral. We know that 1 gram of pyrrhotite contains 0.37 grams of sulfide, and we can use this relationship to report how much pyrrhotite is in the sample if all sulfide is present in the form of pyrrhotite. It is important to know that our method does not yield direct information on whether sulfide is present in pyrrhotite or not. Our method essentially captures all mineral forms of sulfide, some of which are shown in this diagram. All these forms participate in similar reactions and are harmful to concrete, so knowing their total concentration provides a better idea of the reaction potential. In northeastern Connecticut samples analyzed so far, we have seen that all samples contain pyrrhotite only, and none of the other forms. So the assumption that all sulfide is present as pyrrhotite is likely a good one in this region with a particular geology. In addition to measuring sulfide, we also measure sulfate in the sample for 2 reasons. The first one is that we anticipate that the 3 parameters we measure independently should agree with each other. If the relationship that sulfate plus sulfide are equal to total sulfur is true, then we know that our method has little or no experimental error. The second reason is that we can use the amount of sulfate measured to distinguish how much came from the Portland cement in the concrete and how much came from the oxidation of pyrrhotite. We can then compare this to how much pyrrhotite is left to react and estimate the reaction progress in the sample. This knowledge can help us understand the speed at which reactions occurred in the particular location, and ultimately connect it with other parameters that influence that speed. Every sample we analyze brings us closer to understanding the bigger picture.